Hello everyone, hope you are doing good. My name is Arya. I teach biology in Holy Family School. In today's class, we are going to discuss about a beautiful topic called Kingdom Protista. So before going in detail about Kingdom Protista, you should understand some of the basic concepts behind it. Like what is a kingdom or what do the term classification means. So let's go back to what we have studied in your lower class. So before that, let me ask you one question. You have studied classification system, 2 kingdom classification, 3 kingdom classification, 5 kingdom classification, and more about kingdoms, characteristics, examples and everything. It looks so complicated, right? Yes. But why do we classify organisms? What is the need of classifying organism? Yes, it is very important. It is important to classify organism for many reasons. We need to study that organism in detail. How we can study that organism in detail? We can study more about the complexity of their cell structure. Is it prokaryotic or eukaryotic? How is it related to or closely related to other organisms? It is possible only through classification system. So 5 kingdom classification was proposed by R.H. Whitaker, and he classified the organisms into 5 kingdoms. What are those 5 kingdoms? Yes, Kingdom Monera, Kingdom Protista, Fungi, and Kingdom Plantae and Animalia. In today's session, let's see how Kingdom Protista is. Okay, so Protista, so we will see here. We can classify the Kingdom Protista into 3 groups. How many groups? 3 groups. Okay, so let's see what are those 3 groups. So the first one, it is photosynthetic protist. The first one is photosynthetic protist. And the second one, it is slime mold. And the second one is slime mold. And the third one are protozoans. Protozoans. So now we are going to study more details about photosynthetic protist. So before you study any kingdom, it can be Kingdom Monera or Protista or Fungi or Animalia, you should think basic questions in your mind so that you can study all these concepts very easily and you can crack any exams. How do we study? We can understand this concept by thinking few questions. Actually, what is that? How is their structure? That is the first question you should think in your mind. And what is the second? How do they move? Or how do they derive their nutrition? And another question, why or how they reproduce? If you have answer for all these questions, you can easily study any of the kingdoms. Yes, let's see what are photosynthetic protists. So there are 3 photosynthetic protists. They are, first one, that is dinoflagellates. Dinoflagellates. And the second one, it is chrysophytes. It is chrysophytes. And the third one, that is euglenoids. So how easily we can study these 3 photosynthetic protists? Yes, we can compare the characteristics of what we have thought in your mind for these 3 photosynthetic protists. So let's see. So what are the characteristics which we have thought about this? Okay, the first characteristics which we have thought, it is about the structure, isn't it? First we thought like whenever you study any kingdom or any characteristic, anything about any concept, first you should understand what exactly it is. For example, how their structure looks like. Okay, so let's start with the cell wall. The cell wall of dinoflagellates. The cell wall of dinoflagellates, it is made up of a substance called cellulose. Then how about chrysophytes? Chrysophyte cell wall, it is made up of silica. It is made up of silica or silicon dioxide. Silicon dioxide. Yes. And now, how about euglenoids? They don't have cell wall. So, no cell wall. No cell wall. But instead of cell wall, they have a protein layer that is known as pellicle. They have a protein rich layer that is known as pellicle. So, one characteristic is over. Now, what is the next one we have asked? The next one is like, how do they move? Okay. So, the movement, I can write here locomotion. So, how do they move? From the name itself, you can understand dinoflagellates. So, flagella is the structure for the movement. Dino means two. So, they have two flagellae for locomotion. They have two flagellae. Then, how about chrysophytes? Do they have flagella? No, they lack flagella. The next, it is euglenoids. They do have 1, 2, 3 flagella for their locomotion. Okay, so now we understand how their structure is, and we also understood how do they move. Now what is the next question you have thought? Yes, how do they reproduce? So reproduction is the next. Reproduction. So dinoflagellates, it reproduce asexually. How do they reproduce? Asexually. You have already studied there are two modes of reproduction. One is sexual, another one is asexual. Yes. So dinoflagellates reproduce through asexually. And easy tip to remember is, again, euglenoids also reproduce by asexual mode of reproduction. What are the asexual mode of reproduction? Yes, it can be binary fission, okay, or by cyst formation or binary fission. Then how about chrysophytes? Chrysophytes reproduce both sexually and asexually. Both sexually and asexually. Sexually and asexually. This is the next characteristics. Okay. Now let's see one more characteristic. So we have studied now how their structure is, how do they move, how do they reproduce, and let's see how do they derive their nutrition. So from the name itself, you know that photosynthetic protists. What do you mean by that? They carry out photosynthesis. So you can easily say that these all are autotrophs. They carry out photosynthesis in chloroplast. So they all are having chloroplast. So from this, let me ask you one question. Okay. So the one question which I can ask you is, first, chloroplast is present in. Chloroplast is present in. Okay, you have many options here. Okay, the first option is nostoc. The second option is amoeba. The third option is protist, that is euglenoids. Clearly it is mentioned euglenoids. Okay, and the fourth option, that is amoeba, protist, euglenoids, amoeba, then E. coli. Let's talk about E. coli. If you get a question like this, chloroplast is present in what type of organisms? How will you answer? It is very easy. You know that what is the difference between prokaryotic and eukaryotic. So you can easily remember E. coli is a bacteria, isn't it? And it is prokaryotic. Now stock, again prokaryotic. Amoeba and euglenoids are the two options which you can get a confusion here. Both are protozoans. Both comes under protist. Both of these options comes under protist. But in today's session, you have studied what are the characteristics of photosynthetic protist. So who comes under that? Euglenoids. Euglenoid is having photosynthetic mode of nutrition. That means they are having chloroplast also. So the option is C. Okay. So if you understand the concept clearly, you can crack any exams. It is very easy for you to understand, self-prepare the notes so that it is very easy for you to understand any difficult concepts. The most important thing is how you understand it rather than studying, by hearting it. Okay. So let's move on to the next topic. So we will come back to our presentation now. Yes. So kingdom Protista. So here it is, kingdom Protista. So what type of cells they have? They are single celled organisms, some are multicellular, okay, and they are eukaryotic, not prokaryotic, they are eukaryotic. And what are the types of that? We already have studied 3 types. What are the types of kingdom Protista? First one is photosynthetic protist, second one is slime mold, and what is the third one? Yes, it is protozoates, isn't it? So let's see how do they, so where are they found? So next question you should, first we studied what type of cell they have. Okay, they are unicellular, they are eukaryotic. Now we have studied types of protists. Let's come back. Where can we see them? Okay, so they are found as parasites. Some live in water, some live as parasites, some live in marsh, environments. So here it is mentioned some of the parasites, some live in symbiotically in termites. Okay, they live symbiotically. You know that what is symbiotic means, a mutual interaction. They don't harm each other, right? So they live symbiotically in termites. You can see termites here, and wood-eating cockroaches. Okay, this is different from the normal cockroach. Okay, and how do they derive nutrition? How do they derive nutrition? They derive nutrition by a process called a photosynthesis. So that is known as photosynthetic autotrophs, and some are heterotrophs also, that is known as non-photosynthetic heterotrophs. So this is all about the kingdom Protista. So let's see a little detail. How do they reproduce? Next question is, how do they reproduce? They reproduce by sexually and asexually. That is a general one. Characteristic, they divided or they reproduce by sexually and asexually. When we discuss more in detail about the types, it is different from different. For example, for dinoflagellates we studied asexual. Then the other one, which one was chrysophytes, we studied asexual and sexual. Or it depends on the type of the category or the group which it belongs to. So examples you can see here. One example you can see, that is diatoms. Then euglena, slime modes, amoeba, paramecium, plasmodium, all these are the examples of kingdom Protista. Okay, you can see a diagram here, that is nothing but diatoms. See, this diatoms comes under which group? We have studied 3 groups, dinoflagellates, chrysophytes and euglenoids. And this diatom is an example for chrysophytes. Okay, and you can see such a beautiful structure, how their cell wall is made up of. What was their cell wall made up of? Do you remember? Yes, it is made up of silicon dioxide. So this is made up of, the cell wall is made up of silicon dioxide, and you can see that it has got a glassy cell wall. It's like a soap-like arrangement, soap boats. You know soap boats, right? So in that way it has arranged like an overlapping structure. Okay, and it is made up of silicon dioxide, and there is an application behind it. We can use these diatoms. After so many years or so many days, they may die, isn't it? So that time what happens is, the shells of these diatoms, it remains as such, and later it powders and it forms a product called a diatomaceous earth. Let's come back. I will tell you in detail in the next slide. Now let's see the diagram of examples of those protists. Okay, the first one discussed was the photosynthetic protist, which comes in that we studied dinoflagellates. Yes or no? So in dinoflagellates, one of the example we can see here, this is a dinoflagellate, member, and its name is gonialates. What is its name? Gonialates. This actually, it causes red tide. You can see the appearance of C here. See, it causes red tide. It looks beautiful, isn't it? But it is dangerous. How? It is beautiful, it causes red tide, but it releases a poison in the sea which is harmful to the aquatic life. It comes under the photosynthetic protista named dinoflagellates. Okay, So it can appear in different color, yellow, green, brown, blue, red, and bioluminescence also due to the pigment which is present in this. And you can see that dinoflagellates, what we have discussed, how do they move? They move by flagella, isn't it? So how many flagella? Dino. Di means 2. So 2 flagella you can see, one in the transverse, another one in the longitudinal section. Okay. So the next, we have studied about chrysophytes. In chrysophytes, what we have studied in chrysophytes? In chrysophytes, we studied that their flagella, it's not there. It lacked flagella. Now, how their cell wall was? Its cell wall was made up of silicon or silicon dioxide. We studied in the cell wall structure, isn't it? So, some of the examples for this, we can say chrysophytes main examples are diatoms and this is golden brown algae. What is this? Golden brown algae and diatoms. If any question comes for any entrance, any question paper, if the question is like, diatoms comes under which group, you should write it is chrysophytes, and chrysophytes comes under photosynthetic protist. Let's see the next. Yes. So when diatoms die, what happens? You know that its cell wall is made up of silicon dioxide. So when those diatoms die, they leave behind the silica cell wall. So you can see diatoms here. It is the cell wall is made up of silicon dioxide, and once they die, they leave the shell. Okay, the cell wall, which is containing silica. Over billions of years, this had led to the large deposits of silica on the ocean floor, and later it is known as diatomaceous earth. What is this? Have you seen? Over billions of years, the shell, it starts to powder and it forms a product that is known as diatomaceous earth. And there are many uses for this diatomaceous earth. Do you know that? Yes. What is the first use? It can be used for polishing, okay, fine abrasives in polishing. Another one, we can use this to filter beer in breweries, okay, and there are many more applications for this diatomaceous earth. Next, we studied about euglenoids. You can see euglenoids. I will show you the movement of euglenoids video. Okay, so here you can see we have studied that about cell structure. We studied there is no cell wall, isn't it? But instead of that, what do they have? They have a protein rich layer that is known as pellicle. Okay, and then we have also studied flagella. It has 1 to 3 flagella present. And how do they reproduce? They reproduce asexually. This is what we have studied. And let's see, this is a structure of euglena. Example for euglenoid is euglena, and here is the structure. And have you seen one interesting aspect about this? Have you seen a red spot over here? Have you seen? Yes. What is that? Let's see what is that. It has light sensitive red spot. It has light sensitive red spot that is known as, eye spot. What it is known as? It is known as eye spot. And what do they do? It is sensitive to light. Okay, it is because of this eye spot, they can move away from light or they can come towards the light. It is sensitive to light, and it has 1 to 3 flagella. And how do they reproduce? We already discussed binary fission, that is asexual mode of reproduction. Okay, and example is the euglena. Thank you.